Hello, and welcome to the first installment of the 2016 INSAR Summer Institute. I am Carolyn McCormick, co-chair of the INSAR Student and Trainee Committee and a postdoc at Brown University. The INSAR Summer Institute is a 6-week online course presented by the International Society for Autism Research. The goal of the Summer Institute is to provide a forum for the presentation and discussion of autism-related research topics that is accessible for people new to the field. The Summer Institute was originally motivated by and conceptualized for early career researchers, specifically graduate students and postdocs, but is open to all those interested in autism research. We hope that the Summer Institute provides an opportunity for scientists with different backgrounds and from around the world to learn from each other and ultimately advance our understanding of autism. The Summer Institute sessions are free for everyone to attend live and available to INSAR members for replay through their MyINSAR account. This is the second year we're offering this training opportunity, and the theme for the presentations is Familial Aspects of Autism. Today is the first session in our series and features a presentation from Dr. Anthony Bailey, who will be talking about familial risk and comorbidities. Know that there are background materials for today's talk. If you haven't already, you can download these now at autism-insar.org. These materials define some key terms and provide suggested reading in the literature. As with all of the Summer Institute sessions, a group of trainees worked with Dr. Bailey to prepare these course materials, and members of that group will be moderating today's session. Before we get started, know that you can ask questions for Dr. Bailey at any time. We will leave plenty of time for discussion during the back half of the session. You can ask a question at any time by using the chat window on the left of your screen, and we'll get to as many of them as possible during the discussion period. All right, I'd like to get things started by handing the session over to Laura Holmes, the working group leader for this session, who is also a member of the INSAR Student and Trainee Committee. Laura is a doctoral candidate at the University of Utah. She just recently moved to Philadelphia to start her internship at Children's Hospital of Philadelphia. Her research focuses on sexual health and romantic relationships. Thanks everyone for joining us, and welcome Laura.
Hello, welcome everyone. Today we will hear from Dr. Anthony Bailey, Professor of Child and Adolescent Psychiatry at the University of British Columbia. Dr. Bailey investigates the neurobiological basis of autism using genetic, neuropathological, and neuroimaging approaches. At Oxford, Dr. Bailey coordinated the International Molecular Genetic Study of Autism Consortium, a team investigating susceptibility genes for autism. He is the founding editor-in-chief of the journal Autism Research. His clinical work focuses on teens and adults with autism, and his interests include very brief introductions. So without further ado, here is Dr. Bailey.
Thank you very much, Laura. And one very minor correction to the introduction. I'm very pleased to say that David Amaral took over as editor-in-chief of Autism Research last year, which continues to go from strength to strength. And as Rachel and Laura have mentioned, a very warm welcome, particularly to those people who have either stayed up very late or have got up very early to listen to this presentation. So the Summer Institute this year focuses on familial aspects of autism, and this particular presentation is focusing on genetics. And of course, this issue goes closely hand in hand with the final workshop on the broader autism phenotype. So for those of you who may not be able to stay for the entire presentation, I thought I'd start with my conclusions. So the early research into familial factors in autism was very much driven by phenotyping. And that was really the dominant activity until the mid to late 1990s. And since then, the field has really been monopolized almost by molecular genetics. And because of this historical shift, I decided to organize this talk in a chronological fashion. There have been quite a number of surprises in the field. So the mechanisms that can contribute to the development of autism spectrum disorders have turned out to be much more complicated than we initially supposed. there has been gratifying advance in identifying novel genetic changes that contribute to a modest proportion of individuals with an autism spectrum disorder, but by and large these changes are usually associated with intellectual disability. I think a very big surprise has been the very diverse biology that these genes are implicated in. The field began because of a realization that autism was familial, that milder phenotypes could be seen in other family members. But to date, the molecular genetic studies have not really succeeded in identifying the inherited factors in autism. So let's go back to the beginning. We believe that autism has probably always been present in cultures around the world. Rutter and others have drawn attention to historical accounts of individuals who probably had autism, and indeed many researchers have drawn attention to significant historical figures who have contributed much to mankind and probably would have had a diagnosis of Asperger's syndrome. But the first comprehensive account of autism was by Leo Kanner, an American child psychiatrist back in 1943, who assumed that he had identified a disorder that affected children of normal intelligence who had some inborn difficulty in establishing effective contact with other humans. It turned out, of course, that in fact the children that Kanner described did not have intellectual abilities in the normal range. Kanner also drew attention to what he described as personality peculiarities in some of the parents. So by this, he meant that some of the parents were what he called over-intellectualized or cold or aloof, but in his original report made no inferences about these observations. And parenthetically, Kanner was really the first person to observe the association between ASD and increased head size. Now, just 1 year later, Hans Asperger, who was a pediatrician working in Austria, described a somewhat different group of children. So these were able children, usually who had started school, and who were referred to him for conduct difficulties, which were really of two types. Either the children were not getting on with other children at school, or they were getting into difficulties with teachers because they were rather rigid and had their own views about how academic work should proceed. Asperger conceptualized this group of children as having what he called autistic psychopathy. At the time, psychopathy simply meant a personality disorder. And like Kanner, Asperger also observed personality traits in parents that he conceptualized as being related to children's difficulties. And he believed that these personality traits were more marked in fathers than sons, and therefore argued that this was probably a genetic disorder passed from father to son. Now, as many of you will know, Asperger's account was written in German at the end of the Second World War, and it really wasn't until Rutter and Lorna Wing translated and publicized this account that Asperger entered into the diagnostic system. Over the next 10 to 20 years, there wasn't really any advance in genetic thinking about autism spectrum disorder, in large part because Kanner went back on his original belief that autism was an inborn disorder and argued, in keeping really with the zeitgeist in North American psychiatry, that autism was a psychogenic disorder caused by abnormal parenting related to the personality traits that he'd observed. And this trend was really taken to its apogee by Bettelheim, who referred to the mothers of children with ASD as refrigerator mothers. And people familiar with the schizophrenia field will know that mothers were having a uniformly hard time in this point in history because mothers of individuals with schizophrenia were referred to as schizophrenogenic mothers. So there was quite a push in the 60s and early 70s to try and clarify whether Kanner's assumption that there were personality characteristics related to ASD in parents of children with ASD, whether that in fact could be verified. And the early studies, which tended to use general tests of personality, could not confirm that parents of children with ASD were in any way different from members of the general population. Also, in the late 60s, a series of studies, particularly those by Michael Rutter and Linda Lottier, showed that autism was strongly associated with what was then called mental retardation, nowadays intellectual disability, and also strongly associated with epilepsy. And these were key observations that significantly shifted the field away from the idea that autism might be a psychogenic disorder. At that point in time, the early epidemiological studies suggested that autism only affected about 2 to 4 in 10,000 children. So one can see that there's really been a dramatic increase, particularly in the last 15 or 20 years, in the number of children identified with ASD. At that point in time, early studies suggested that the recurrence risk in autism was of the order of 2%, and several authors argued that because of this low recurrence risk, it was unlikely that genes contributed to the risk for autism, until it was realized that a recurrence risk of 2% was 100 times greater than the population prevalence of 2 in 10,000. So it was the increase in recurrence risk, in other words, if one has one child with an autism spectrum disorder, one's at increased risk of having a second affected individual, that led to the first twin study. And twin studies are necessary in order to determine whether increased familial risk is conveyed by environmental or genetic factors. So Susan Folstein from the US and Michael Rutter from the UK carried out a study in the UK in which they attempted to identify all the same-sex twin pairs in which one of the twins had a diagnosis of autism. They chose same-sex twin pairs because of the difference in the incidence of autism between males and females. What Folstein and Rutter found was that in 36% of the monozygotic or identical twin pairs, both of the twins had autism, whereas in the dizygotic or non-identical twin pairs, always only one twin had autism. So this very large difference in concordance for autism between pairs which are genetically identical compared with pairs in which both individuals on average share only 50% of their genes indicated that autism arose on the basis of a very strong genetic predisposition. Folstein and Rutter also observed, however, that the majority of the non-autistic identical co-twins had some sort of cognitive difficulty. That's these individuals here, which principally seem to relate to language difficulties. And so their hypothesis was that the genetic risk extended to a broader range of difficulties than autism as narrowly defined. We jump ahead a little bit. Christopher Gilbert's group carried out a very comparable study in 5 Scandinavian countries and replicated the very striking difference in concordance rates between identical and non-identical twins, but really didn't find much evidence for the presence of related difficulties in non-identical co-twins because in identical non-autistic co-twins because the concordance rate in their twins was so high. What Folstein and Rutter were able to conclude was that the heritability of autism was very high. And what heritability means is the proportion of individuals in a population who have a disorder because of a genetic basis. They were also able to conclude that the underlying genetic mechanisms could not be Mendelian because that would not fit the observed concordance rates, and that therefore autism must arise on the basis of the interaction or the role of several different genes. Also, they emphasized that autism did not seem to be a deterministic disorder. In other words, if one inherited the genes that predisposed to autism, that is all they did. They didn't determine that you would develop autism, and this is often referred to as variable phenotypic expression. Now, if we jump ahead to the ensuing couple of decades, there were several significant developments. One was that various different groups were conducting family history studies to try and learn more about the recurrence risk for autism and also to try and clarify this issue of quite what it was that the genes predisposed to. Was it autism as narrowly defined, which might have been a conclusion from the Scandinavian twin study, or was it a much broader range of difficulties, and if so, what were the characteristics of these difficulties? Also, in the Folstein and Rutter twin study, there was no standardized diagnostic interview, and it was really the need for standardized diagnostic interviews, particularly for genetic studies, that led to the development of the ADI and the ADOS, spearheaded by Michael Rutter, Kathy Lord, and Anne Lecuter. The follow-up twin study that led by Anne Lecuter and Michael Rutter basically confirmed the strong genetic predisposition to autism, but also indicated that the risk extended to milder social difficulties, as well as to the language-related communication difficulties picked up in the first study. And the best of the family history studies at the time in the UK, led by Patrick Bolton and Michael Rutter, and in the US by Susan Folstein and Joe Pivin, estimated that recurrence risk was now 6%. So there was much work at this time in trying to characterize the broader autism phenotype. It seemed that social and communication difficulties frequently co-occurred, that these difficulties were seen in the relatives of singletons with autism and therefore couldn't be attributed simply to being a twin, and that these milder problems related to ASD were more common and more severe in male than female relatives, following the pattern seen of risk in individuals with ASD. What was also important was those characteristics that did not seem to be part of the broader autism phenotypes. So relatives, by and large, were not showing intellectual functioning in the below average range unless they also had ASD, and there was no obvious increase in the risk for epilepsy in individuals who did not have the full-blown disorder. So at the time it was thought that the characteristics such as mental retardation, epilepsy, or indeed loss of language are what are sometimes referred to as emergent phenomena. In other words, they're only seen when the disease is expressed to its fullest extent. It was also possible to conclude that if characteristics related to autism were present in other relatives, that at least some of the genes that were involved, particularly in parents, must be dominant genes. Otherwise, one would expect to see two unaffected parents having a child with autism if all the genes were recessive in nature. Many of the family history studies also looked at the characteristics of relatives in terms of the presence of other psychiatric disorders, and there was some unanimity in finding elevated rates of anxiety, depression, and OCD in the relatives of people with autism. And what was particularly striking was that this increased risk for psychiatric disorder could occur in individuals who were not living with the proband with ASD, suggesting that these difficulties were not a consequence of any stress associated with having a relative with ASD. At the time, there was not a great deal of interest in comorbidities in individuals with ASD. So it's really only in the last 10 or 15 years that we have become aware that individuals with ASD are prone particularly to anxiety and depression and OCD, and to a lesser extent eating disorders and psychosis. And it's also worth noting that the early family history studies were not paying attention to the presence of ADHD in other relatives, even though we know that is a strong comorbidity with ASD. So the conclusions from the twin studies and pooling the family history studies was that autism could now be considered a common disease to a geneticist. A disease that affects more than 1 in 1,000 of the population is referred to as common. The best modeling of the possible underlying genetic architecture that was carried out by Andrew Pickles using the UK twin and family history data suggested that probably at least 3 genetic loci needed to be involved, and one couldn't rule out the possibility that as many as 10 loci could be involved in incurring risk for ASD, and that probably there was what was referred to as epistatic interaction between these loci. In other words, they didn't act in an additive manner, but one needed a particular combination of genetic loci. So many groups in the mid-90s, on the basis of the evidence from these careful phenotypic studies, advanced to molecular genetic studies of autism. And the reason for doing this is particularly related to the third bullet point, which is that if one can identify genetic risk factors, we can then deduce what proteins are involved, and this could shed some light on the neurobiology of autism, which both then and to some extent now remains relatively unknown. Also, if particular genetic risk factors could be identified, this might lead to much more accurate genetic counseling in individual families rather than us having to give families a population-based recurrence risk estimate. And of course, sometimes genetic risk factors for autism might convey risk for other types of medical or psychiatric disorder, but it can be helpful for clinicians to know about. So I'm going to spend a couple of minutes just reminding the audience a few basics about genetics. So genes are really what convey the blueprint for the different constituents of cells and the way that cells interact with each other to form organs and of particular interest to us, the brain. And you will all recall that genetic material is carried in structures called chromosomes, and historically the only way to identify genetic change in individuals was if it was at a chromosomal level and could be picked up using special staining in a technique known as karyotyping. The genetic information itself is conveyed by the particular sequence of base pairs in the molecule DNA. And so this is an example of a stretch of DNA, and usually stretches of DNA of the order of hundreds to tens of thousands of base pairs convey a code for a protein. This very large amount of very long molecule somehow has to be packaged into the nucleus of each cell, and this is done by wrapping the DNA in a very tight manner into the chromosome, and particularly important in this regard are structures called histones, which are involved in the packing of DNA and also the unraveling and turning on and off of different genes. We've known for a very long time that it's the sequence of three particular base pairs that codes for an amino acid. This is a redundant code. The U in this particular diagram is because the diagram relates to bacterial DNA. And so there are several amino acids for which there are redundant codes, but of course one can also have stretches of base pairs that do not code for any amino acid at all. And so the cell has mechanisms for reading off the sequence of DNA, building up and linking to each other a chain of amino acids, which as it gets longer tends to start to fold and acquire secondary and tertiary structure, so that many of the proteins which make up our bodies really have extremely complex overall shape. And it's worth emphasizing that changes in the amino acid coding sequence very often lead to changes in the function of protein because they affect protein shape, and protein shape affects how proteins can bind to other proteins or other types of molecules. Now, it's not simply the case that DNA codes for genes in a very simple way, so genes have very complex internal and external structures, so they have areas outside of the coding region that are controlling the turning off and on of genes that initiate the process of reading off the DNA code. And within genes, most genes are split into exons, which is the sequence of DNA that conveys information that translates into a protein, separated by introns, which are stretches of DNA which are not involved in coding for protein structure. So this is overall a very complicated arrangement, and we continue to learn information about the fine detail of this process, and particularly the mechanisms of initiating the turning on and off of genes. So, having taken that slight detour, let's get back to our timeline. Although most cases of autism spectrum disorder are what we refer to as idiopathic, in other words, we do not know the identity of the underlying genetic cause, we have known for quite a number of years that autism can also arise on the basis of single gene disorders, of which the most common are Fragile X and tuberous sclerosis. Approximately 20 to 40% of people with Fragile X, males with Fragile X, that is, develop an autistic phenotype, and about half of individuals with tuberous sclerosis, even though both of these disorders only account for a very small percentage of the total number of individuals with ASD. There's been much work in identifying the role of the protein that is reduced in amount in Fragile X, and the mechanisms at a cellular level that tuberous sclerosis interferes with are increasingly being worked out, and that has been a continuous process since the 1990s. Also in the 90s, there was an increasing recognition that autism could be associated with a very wide range of chromosomal abnormalities, although there was no particularly obvious pattern to that association noted at the time. But a particularly important development, and really this is now a theme from this point onwards, was that technological developments in laboratory equipment enabled individuals to start to study the genetic basis of many different types of disease. Prior to this point in time, it was extremely time-consuming to identify genetic risk markers, as this had to be done using radionuclide technology, which took a long time and was very labor-intensive. So the development of machines that could automate marker detection was a significant advance and enabled people to carry out studies of large numbers of individuals with different medical diseases. It's important to note, however, that at this point in time, the human genome had not been sequenced. And so the machines were working by identifying what used to be referred to as microsatellite markers, which are stretches of essentially junk DNA that are inherited in a stable manner akin to the inheritance of genes. Because there was very little idea at this point in time about what the biochemical basis of autism might be, in other words, there were no obvious candidate genes, most groups decided to conduct what were called affected relative pair linkage studies. These involve finding families that contain two or more individuals affected by a disease and looking for areas of the genome using markers where the two affected individuals, when they're all pooled together, seem to share that structure of the genome more than 50% of the time, which is what would be anticipated if that area of the genome had no role in the development of the disease. If that area of the genome is implicated in the development of the disease, then in a rare disorder, two affected siblings are probably carrying genetic risk factors in that area, and therefore sharing would be greater than 50%. And a small number of groups did study positional candidate genes, and this refers to genes that become candidates because they're close to or in the area of chromosomal breakpoints in in those individuals who have a chromosomal abnormality and autism. So there were a large number of studies in the late 90s and early 2000s using genetic linkage approaches and an affected relative pair design. This was a slide I made in 2002, and the stars indicate where different research groups were finding evidence of linkage on different chromosomes. And what one can see is that there are some areas of some chromosomes where linkage was identified by multiple groups. And so there was a lot of hope at this point in time that by fine mapping these particular areas where different groups and different samples had found evidence of genetic linkage, that we would quickly identify autism susceptibility genes. In fact, that turned out not to be the case, and the explanation for that is quite interesting. So this is an example of the linkage peak on chromosome 7 that was one of the first identified peaks, and one can see that this is really a very broad peak, and the diagram here is spanning about 300 genes. And the reason the peak is so broad is because of the extreme genetic heterogeneity of autism. In other words, there are many families who do not carry risk genes in this area, and therefore they are spreading out the area of linkage. There have been some positive successes in terms of using this approach. So the gene MET has survived all sorts of different analysis, but this didn't deliver the type of expected gains that we had hoped for. Of course, people tried to reduce heterogeneity by examining subsets of relative pairs on the basis of some phenotypic characteristics, such as whether individuals had intelligence quotients in the normal range or did not have a history of language delay. But these approaches were not terribly successful. And it's worth noting that overall there was very little evidence of linkage on the X chromosome. So if we now move into the new millennia, several interesting developments, of which probably the most important was continuing development in genotyping technology that initially enabled us to identify so-called single nucleotide polymorphisms. So these are changes in a single base pair in a gene that would lead to a change in an amino acid or a failure to produce an amino acid, and much more recently the capacity to sequence either exons or genes. Additionally, this pattern or lack of specificity to the chromosomal abnormalities associated with autism gradually seemed to take more shape, so that particular chromosomes seem to carry a particularly high risk for the development of autism, and these are some of the more common chromosomal abnormalities associated with ASD. But a striking feature of these abnormalities is that they show extremely variable phenotypic expression. So although some individuals with the abnormality may have ASD, others may simply have intellectual disability or language problems, or indeed sometimes some other psychiatric disorder such as schizophrenia. The other notable finding was that the study of chromosomes did not really reveal any association between autism and subtelomeric deletions, which are a known risk factor for intellectual disability. Also, people continue to devise better measures of the broader autism phenotype, and you will be hearing more about that in the final lecture in this series. The early studies had all taken a linkage approach to autism, and that essentially is asking the question, where on chromosomes might susceptibility loci be identified? But we have had a whole series of association studies which ask, are there particular variants of a gene that show a strong association with autism? And there have been some positive findings from those studies, but it's fair to say that the expectation from the mid-90s that we would find susceptibility loci that were commonly causing autism in a large proportion of cases was not supported by any of the association studies. And quite unexpectedly, in parallel with this work on the genetics of autism, several groups found an association between the risk of autism and age of parents, usually the father, but particularly for some of the chromosomal abnormalities, an association also with maternal age. And the usual interpretation of this sort of association is that with increasing paternal age, there is an increased risk of mutagenesis in parental sperm, and that can increase the risk of developing a disorder. It's also possible, of course, that individuals who may have milder autism-related phenotypes may delay marrying and reproducing, and of course it's possible that both mechanisms might be active in the same individual. So a lot of excitement was generated about 7 or 8 years ago when several groups identified risk genes for autism that affected synaptic function. And it was Thomas Bergeron's group in Paris that kicked off developed this particular development, finding changes in individuals who had chromosomal abnormalities on the X chromosome that turned out to be linked with neurexin and neuroligin genes. This development and subsequently the implication of the protein SHANK, also involved in the synapse, generated a lot of interest, in part because a lot is known about synaptic function, and of course many of the drugs for psychiatric and neurological diseases target synaptic function, and so this opened up the possibility that if autism turned out to be a disorder of the synapse, that it may be possible to develop effective biological treatments. And almost simultaneously, a new genetic mechanism predisposing to autism was identified. And this became possible because of the change in technology in the laboratories that enabled us to type single nucleotide polymorphisms, revealed that some individuals carry stretches of DNA where there is a variable number of copies of that stretch. In typically developing individuals, we all have about 200 regions in our chromosomes where we have variable numbers of repeats of a stretch of DNA that can range from a relatively small number of base pairs to thousands of base pairs in length. And there was a realization that some of these copy number variants that were being picked up in autism overlapped either with chromosomal abnormalities identified through karyotyping studies and subsequent fine mapping, or they overlapped with genetic change picked up by other techniques. And so there's been a huge amount of work over the last 10 years to try and characterize further these copy number variants with a particular focus on de novo copy number variants. So these are the ones that are associated with autism but are not passed on by parents. And it's been encouraging that particular areas of the genome have been identified in which there are changes in copy numbers, variants in different samples, that they overlap with mutations, that the size of these copy number variants tends to be larger in autistic populations than in the rest of the population. But if one looks at particular loci, one sees that the copy number variants are actually behaving in a very similar manner to other genetic risk factors, in that often the phenotype associated with the copy number variant is very variable, that there are affected relative pairs in which, or there are sibling pairs in which both individuals carry the same copy number variant, but only one of the individuals has ASD, or conversely, there are affected relative pairs in which both individuals have ASD, but only one of them carries the individual copy number variant. So it seems as though copy number variants are a contributory mechanism to autism, possibly acting in concert with other as yet unidentified risk factors, but that probably in the majority of cases an individual copy number variant may not be sufficient cause for ASD. And we think that copy number variants convey risk either because they interfere with a single gene or are interfering with multiple genes, or possibly unmask a recessive allele on the other chromosome. And particularly interesting and an area of increasing study is that genetic change, for instance, conveyed by a copy number variant, may exert effects on the turning on and off and transcription of genes at some distance away from the insertion of the variant itself. So I mentioned earlier about association studies, and I just want to pick up one particular finding that was first brought to attention through association, which is the association between ASD and a gene called P10. This is one of the more relevant associations clinically. P10 is what's known as a proto-oncogene and conveys a risk for the development of cancer. And various groups have found that if one studies individuals with autism who also have macrocephaly, that is increased head circumference, then about 2% of those individuals will carry a change in the P10 gene, which should be identified because of the associated risk of cancer both to that individual and anyone else in the family who carries that particular variant of the gene. Now, most recently, the literature has been dominated by papers in which groups have used the capacity to sequence the genome, either to sequence exomes of genes, the coding regions of genes, or indeed to sequence the whole of a gene, which includes those areas of the gene that are responsible for turning the gene on and off. And yet again, a new genetic mechanism has been identified as the technology improves, so that we now know that some people with autism carry a very small insertion or deletion of DNA, which is abbreviated to the term indel, which in some individuals seems to confer a risk for autism. We've also seen very innovative use of different types of high-risk samples. So Chris Walsh's group in particular has taken advantage of high rate of consanguinity in some populations to use these consanguineous families to identify recessive alleles that predispose to ASD. And more recently, Chakrabarty's group have published a nice paper in which they focus on severely affected females with ASD, the rationale being that because females are the less frequently affected sex, that they may need a higher genetic load to develop ASD than do males. So we're in a position where at the moment over 100 genes that potentially confer risk for ASD have been identified. The next slide simply shows those that until recently were considered some of the top genes, particularly on the basis of single nucleotide variant or polymorphism studies. What's striking about these genes is that they are involved in a very wide range of biological processes. So some of the mechanisms that have been implicated are synaptic, as I previously mentioned. Recently, chromosome modification gene involved in modifying chromosomes in a way that affects the ability of genes to be turned on and off. Genes involved in regulation of transcription of other genes, and then a wide variety of genes involved in cell adhesion, axonal projection and pathfinding, immunological function, and genes that seem to be targets of the Fragile X protein, and other genes related to Wnt signaling and the mTOR pathway. So I'm moving into the final section of the talk, which really involves summarizing where we are and some thoughts about where we should be going next. There's no doubt that the number of different mechanisms that can lead to ASD at a genetic level has turned out to be a surprise, and this is not unique to ASD. Much of what I'm saying would be equally applicable to the fields of schizophrenia or ADHD. There have been a lot of successes in identifying new mutations or new chromosomal abnormalities or new copy number variants that seem to confer a high risk for the development of ASD. But it's also fair to say that probably the majority of these changes are also associated with intellectual disability. There have been several attempts to pool genes, genetic information and phenotyping from a wide variety of psychiatric disorders, and what that has revealed is that there are some genes that seem to confer a modest amount of risk for a number of apparently unrelated disorders. But if we put all of this information together, we have still probably only identified genes that account for perhaps 10 to 20% of cases of autism. And none of the identified genes would be what are usually referred to as genes of major effect. In other words, that they're conferring a very large risk, but also in a significant number of people. What we have not achieved so far is identifying genes that seem to be involved in the inheritance of ASD. In other words, that genes that are associated with the broader autism phenotype in other family members, which therefore should be passed from parent to offspring. And again, this is not a unique situation for ASD. The same situation arises in schizophrenia and bipolar disorder, and sometimes this is referred to as missing heritability. In other words, molecular genetics studies have not identified sufficient genetic loci of significant effect to account for the observed heritability. And finally, none of the genes that have been implicated in autism so far are really able to account for the significant sex difference in the disorder. So what are some possible ways forward? I decided to group these under 4 headings: epidemiology, neurobiology, genotyping, and phenotyping. So a lot of work has gone into carefully collecting and phenotyping large samples of individuals with ASD. Sometimes these are affected relative pair samples in which 2 individuals in a family are affected, and sometimes, as in the Simon-Singleton collection or simplex collection, these are families in which only 1 individual is thought to have ASD. But these samples all vary tremendously from one to another, as one can deduce if one reads the characteristics of the sample. They differ in terms of average IQ, the association with epilepsy, the rates of language delay, and so these research samples are all biased in slightly different ways. Additionally, many of the samples tend to have a strong representation of people with ASD and comorbid intellectual disability, whereas most recent epidemiological studies suggest that the majority of people with an autism spectrum disorder nowadays have intellectual abilities in the normal or average range. So we're left with some uncertainty as to the extent to which the particularly de novo changes identified in research samples would be relevant in epidemiological samples. And so there's a clear need for several independent epidemiological studies of ASD to establish the role of the identified changes in the general population, because this is particularly relevant for informing decisions about genetic testing of children and adults. At the moment, it would be normal to carry out some sort of genetic testing of individuals with ASD who were dysmorphic or had intellectual abilities below the average range, but great uncertainty as to the likely hit rate of such testing in more able individuals. And epidemiological studies can be very informative in telling us what sort of protocols we should be following. I've drawn attention to the very diverse nature of the neurobiology implicated by the identified risk gene, but bioinformatics experts have published numerous papers showing possible ways in which the proteins from some of the identified genes may interact, and we can expect much more of this type of analysis, which can lead to targeted genotyping in pathways that seem to be overrepresented. And there's been some suggestion recently that some of the genes implicated in autism may be targets of the FMRP protein, the protein related to the Fragile X gene, or mTOR, a protein that's strongly controlled by the genes implicated in tuberous sclerosis, or very recently, the suggestion that many of the genes implicated to date in ASD may also be targets of the chromatin modifier gene CHD8. Some people in the audience might be wondering, why would a chromatin modification gene be implicated in autism? And one possible explanation is that the effect is indirect, in that it is in some way altering the turning on or turning off of genes implicated in ASD rather than having a direct effect. In terms of genotyping, I think it's likely that we will see increasing focus on the non-coding regions of genes and the mechanisms that underlie gene regulation, as we so far have not been very successful at identifying genes contributing the bulk of risk, which leads to the possibility that what we should be looking for are not changes in protein sequence, but in the amount of protein that is produced, and that would be controlled by the turning on and off of genes. The Y chromosome is a particularly difficult structure to analyze genetically, and there is still work to be done there. But also, we need to shift, I think, some of our genotyping focus away from individuals with lower intellectual functioning to the more able end of the autism spectrum. And again, we could utilize the strategy of particularly focusing on able females. Now, the field of autism began really with Kanner and Asperger drawing attention to the existence of milder and presumably related phenotypes in other family members. But there have been very few large studies in which there has been a comprehensive attempt to phenotype parents and siblings of individuals with ASD using measures of the broader autism phenotype and factoring that phenotypic information into the genotyping analysis. I think the reason that has not been done is because it's very costly in terms of identifying and assessing these families. But if we really want to make progress in understanding the inherited components of ASD, then that is an issue that needs to be tackled. And of course, we could continue to try and dissect out the autism phenotype by taking affected relative pairs and particularly focusing on individuals who were discordant for a particular feature, whether it was intellectual disability, language regression, or epilepsy, in case there are risk loci that contribute to particular subcomponents of the phenotype. Now, we have seen a lot of chromosomal copy number variant and new mutations associated with autism over the last 5 to 10 years. But in only a few of these cases has there been very careful phenotyping of individuals who carry the variant. And that seems to be important because it may be that particular variants confer risk for particular types of autism in terms of the phenotype that the person shows. Similarly, it's possible that genes that are involved in particular interacting protein networks might predispose to types of ASD that show some common phenotypic characteristics. But we really don't have sufficient detailed phenotyping of these changes to know whether or not that would be the case. I've already alluded to the need to measure the broader phenotype in samples for genotyping, and this is particularly important in ASD because of something that the field has tended to ignore, which is a process called assortative mating. In other words, we do not select our mates randomly, we select them on the basis of characteristics that are appealing to us, or compatible with us, and therefore it's possible that people who carry risk genes for ASD for various reasons might choose to marry each other. And Joseph Piven has very sensibly pointed out that a useful strategy might be to focus on families in which we believe one of the parents has the broader autism phenotype and the other does not show any obvious signs of autism-related difficulties, because that way it would give additional power to association studies. And finally, in terms of phenotyping, although there has been numerous attempts to increase the homogeneity of samples by focusing on characteristics of autism such as intellectual disability or language delay, there's been relatively little interest to date in looking at stratifying samples by the presence or absence of epilepsy or other comorbid psychiatric disorders, and that might be a useful way forward. And my final slide really relates to the implications of the findings to date for our understanding of ASD. If we go back to when different groups began molecular genetic studies in the mid-90s, the underlying assumption was that we would identify genes involved in a very small number of biological processes leading to autism. In other words, that autism was probably a specific disease where we would be able to pin down the neurobiology quickly. And of course, that has not turned out to be the case. Now, of course, it could be that in the overwhelming majority of people with ASD, where we have not identified genetic mechanisms, that may turn out to be what we discover. If we do not discover that, then it would seem that the way to think about ASD may be much more similar to the way we think about intellectual disability. So we have no difficulty in working with the concept that intellectual disability can arise on the basis of hundreds, if not thousands, of different genetic risk factors, each of which only contributes to a small proportion of the whole of intellectual disability, and that the unity of intellectual disability is how those different genes impact on cognitive processing. So it may be that many individuals with ASD do not share common neurobiological features, and that the unity to the syndrome arises at the impact of the genes on systems biology or cognitive processing. So I believe my hour is up, and I'm now going to hand over to our moderators, Laura and Rachel, who've been paying attention to the questions that have been asked whilst I've been speaking. And now is the time, if you have questions, to type them into the chat box.
Thank you so much, Dr. Bailey, for a great presentation. And as you mentioned, if you have remaining questions or if questions come up during the discussion, you can still ask them in the chat feature of ReadyTalk. But now I would like to introduce Rachel Earle, the member of the sessions working group who will be moderating the question and answer session. Rachel is a graduate student at the University of Washington in the School of Psychology program. Take it away, Rachel.
Wonderful. Thank you, Carolyn. And thank you so much, Dr. Bailey, for the presentation. I want to thank our audience for great questions throughout the presentation. We have a lot of questions, so we'll go ahead and get started. All right, so Dr. Bailey, thinking back to kind of the beginning of your presentation when you were discussing the history of ASD, we have a few questions related to that. One question was regarding the increased risk of autism today compared to what it looked like earlier in the 1900s. One member of our audience was asking whether that increased risk is due to advances in diagnosis or other factors, and what your thoughts are on that increase.
So to recap, we have gone from a situation in which the accepted prevalence of autism was 2 in 10,000 to it now being greater than 1%. So in British Columbia, where I work, the rate last year was about 1 in 70 children. If one looks at the characteristics of identified children over time, we can see that there has been a shift from historically 3/4 of people with ASD having intellectual impairment to nowadays roughly 3/4 of individuals with ASD not having intellectual impairment. So we believe that a huge factor in the increase in detected prevalence is that we are now identifying more able individuals who historically would not have received a diagnosis of ASD. My clinical work nowadays is almost exclusively with adults, reflecting some unmet need where I work, and I continue to see individuals every week in their 20s and 30s and 40s who have ASD previously undiagnosed associated with normal intellectual ability. So I think it's likely that we will continue to see the identified prevalence increasing over the next few years. Certainly in BC, we have comparable data from each of the last 7 years, and there doesn't seem to be any sign of that graph plateauing out. Of course, we can't exclude the possibility that there has also been a genuine real increase in the rate of ASD, but that would be a very hard issue to really clarify with any great precision at the moment.
Thank you, Dr. Bailey. Another question is in relation to broader autism phenotype, and questions regarding whether is the broader autism phenotype considered pathological, and is it diagnostically classifiable?
So the broader autism phenotype also ranges in severity in the same way that the core disorder ranges in severity. So some individuals can have a history of language delay that in adult life can continue to be associated with spelling and reading difficulties. Some people will have social issues which tend to lead them to avoid social interactions, or other people may have poor social judgment that can cause some difficulties in everyday life. So some individuals with the broader autism phenotype, it does cause difficulties, and others it causes no difficulty at all. And the broader autism phenotype is not necessarily a bad thing, in that we know that many people who show some of these characteristics are extremely successful individuals who can often show incredible amounts of determination and persistence. And so one way of thinking about the genes that predispose to autism is that in some individuals they may be associated with very positive traits, so that in families where we're dealing with an inherited form of ASD, these genes or alleles may have persisted in the population because they confer significant advantage.
Thank you.
Oh, and I realize I didn't add that the broader autism phenotype is not a recognized diagnosis either in ICD-10 or DSM-5.
Thank you. Given your clinical background, when adults with autism who are thinking of becoming parents ask about risk, what do you tell them? And is genetic counseling helpful in your experience to recommend?
That's an interesting question, because actually I can't think of a single couple where one of the individuals has ASD where they have been thinking of starting a family and have asked my advice about whether they will have a child with autism or not. I think most of those couples have worked out for themselves that they have an increased risk. I do see many mothers each year who have a child with ASD, and it is only after the child is diagnosed with ASD that the mother realizes that she may also have the disorder and seeks a diagnosis. I don't think we're in a very strong position to give people very accurate probabilities for their chance of developing ASD. Of course, people who want the best information should go to clinics where they can receive genetic counselling, which may first have to be preceded by genetic testing to see if either of the parents carries a currently identified risk locus for autism.
Thank you. Given issues of accessibility and insurance coverage of genetic testing for families, how is that being addressed in the clinical community?
I can only speak for British Columbia, and the situation may be different in the USA. At the moment, I'm not aware of any insurance company here paying any attention whatsoever to risk genes for ASD in making assessments about insurability.
Do you feel like that's an area of further work?
Well, there's divided opinion about this. If you're an insurer, you want as much information about risk as possible because that enables you to set premiums for liability, for instance, for earthquake or natural disaster or financial impecuniety or specific disease if you have very specific information. An opposite view is that that logic works fine for earthquake and flooding, but that people should not be penalized for their genetic makeup because they had no choice in the genes that they were handed.
Thank you. What are your thoughts regarding the therapeutic and treatment implications of genetic studies of ASD?
So one of the reasons I drew attention to the great excitement caused by the identification of mutations in neurexin and neuroligands and shank was the possibility that this could lead to drug treatments for ASD, and indeed there have been various trials of drug treatment for Fragile X, tuberous sclerosis, Rett syndrome. At the moment, I can't see that there is a locus that has been implicated in a sufficiently large number of people that this would be an economically sound prospect for a drug company to engage in new drug development. However, it is the case that quite often people find new uses for existing drugs, many of which affect multiple biochemical pathways. So of course it's possible that there may be some individuals with ASD that have an impairment in just one specific biochemical system where there may be an existing drug that might affect that. But I think we really need to wait and see how things advance in the majority of individuals in whom we've not yet identified genetic risk factors. If it turns out that a very small number of genes account for the majority of risk, then that would be an attractive proposition for drug development. If it turns out that in those 80% of individuals where we've not yet identified genetic risk factors, that the risk factors turn out to be as heterogeneous as those so far identified, and that we can't identify any commonality in terms of protein pathways, then drug development would be a long way off.
Mm-hmm. Thank you. Given your discussion of synaptic dysfunction, a question related to that, can synaptic dysfunction be considered as a central mechanism for pathophysiology of ASD? And can the mechanism also explain intellectual delay, disability, and potentially be a converging mechanism that could explain that comorbidity?
So I think what I need to emphasize is that the genes that have been identified that are involved in synapses only seem to be implicated in a tiny percentage of individuals with ASD, and so it's premature, I think, to extrapolate and describe ASD as a synaptic disorder. Of course, we know that genes such as two genes implicated in tuberous sclerosis impact on synaptic functioning. And it's fair to point out that most of the identified genes in ASD that are involved in synapses are found in individuals who have comorbid intellectual disabilities. So it's by no means clear that in individuals with intellectual functioning in the average range that autism is a synaptic disorder.
Thank you. What do you think about computational or statistical machine learning methods for detecting important genes associated with ASD?
I don't think a great deal about them because they are beyond my cognizance, by which I mean I don't really understand the inner workings of those very complicated mathematical models. So for people who are as ignorant as me, the idea here is really to feed in bioinformatic information in terms of identified risk genes, the proteins they code for, and then allowing the machine to hypothesize which protein pathways may be implicated, and also, particularly importantly, to hypothesize new genes that should be tested. Because I'm not a statistical geneticist, I'm not really able to comment on the merits of one approach versus another and how difficult it is to disprove any findings. By and large, the acid test in genetics is whether different groups using different samples can come up with the same findings. So any prediction from these machine learning algorithms is usually derived from one data set, and to really know whether that is meaningful, the prediction needs to be tested in an entirely different group of families.
Thank you. How would you summarize the current genetic explanation for autism being a male-predominant disorder? And how do we understand the genetic etiology in females versus males?
So when people started doing molecular genetic studies, it was really quite unclear whether genes would be identified on the X chromosome that explained the bulk of the difference in the sex ratio in autism. And it's turned out that very few genes on the X chromosome have been identified, and these account for really a very small proportion of cases. So the sex difference in autism is still a mystery. Historically, we've tended to assume that sex differences, if they're not due to genes on the X chromosome, are probably caused by differences in levels of sex steroid hormones during development. But more recently, there's been interest in whether there is sex-specific expression of genes during development. The idea that females may have a greater genetic loading really stems from the very old multifactorial threshold model of complex diseases. And that still really is a hypothesis at the moment. So the work that I referred to recently by Chakrabarty focused particularly on very handicapped females with ASD. We really don't know whether the genetic architecture in very able girls and women with ASD is different or identical to that in equally able males. So this is still a big unknown.
Thank you. What is the current common misconception surrounding the genetics of autism that you would like to correct or clarify?
So sometimes I hear people talking about genes for ASD. In other words, there is a one-to-one relationship between inheriting the gene and developing ASD. And actually, that is true in really a very, very tiny fraction of individuals. Of course, it is those individuals where we identify the genes that make the scientific papers, but overall the single gene causes or copy number causes that are sufficient cause are in the small minority. And I'm sorry, Rachel, I blanked on your question. Can you just remind me?
Oh, no problem. Just a current common misconception that you would like to correct surrounding genetics of autism.
So genes are better thought of as being risk factors for autism. In other words, we still believe that in most people probably more than one gene is implicated. All the evidence we have is that even in identified genes, there's still extreme variable expressivity. So even if one carries a risk gene, it's not inevitable that one will develop autism. One could develop some other sort of difficulty or indeed have no difficulty at all. And I think sometimes I've heard people for a variety of psychiatric disorders refer to good genes and bad genes. There are no good genes or bad genes. If autism has persisted over millennia because of genes that are conveyed from one generation to another, then those genes, when they are not in the setting of other risk genes, must confer some advantage to the individual. So it's not helpful, I think, to think of genes as being good or bad, and there are lots of reasons for thinking that genes associated with autism might indeed confer considerable advantages relatives who carry the genes but don't have the full-blown disorder.
Thank you. We have a couple questions related to comorbidity in autism, and one of the questions relates to the comorbidity between ASD and OCD, as well as it's been seen in clinical studies that in mothers of children with ASD, OCD can be common. And what are your kind of thoughts on that comorbidity and the potential research that's needed in that area?
So I think two issues here. One is OCD is a very common comorbidity in ASD in childhood and in adulthood. And in fact, all the evidence we have is that anxiety, depression, and OCD as the major psychiatric comorbidities increase in prevalence in adult life compared to childhood. So in adulthood, sometimes clinicians are spending most of their time managing the comorbid disorders rather than the consequences of ASD itself. It's also the case that both clinically and in family history studies, we see elevated rates of these comorbidities in relatives. And so when I'm trying to explain this to families, I say it's quite possible that some of the risk genes for ASD why in one of my closing slides I suggested that it might be beneficial in the future to do some analysis of existing or new data sets according to individuals grouped on the presence of these psychiatric comorbidities.
Thank you. Is there emerging research that you know of regarding undiagnosed or late diagnosed females and comorbid borderline personality disorder?
So, just for the benefit of the audience, by and large, the sex ratio in autism is generally thought to be 3 or 4 males for every female. But over the years, there have been a number of epidemiological studies of Asperger's syndrome, which have found sex ratios as high as 8:1 or 15:1. Now, typically, we don't usually in medicine think that sex ratio varies across the spectrum of severity of disease. And I think what has happened recently is that people have shifted to the view that we are simply not very good at identifying the most able females with an autism spectrum disorder. So I pointed out earlier that every year see 2 to 4 women who are first identified after they have a child with ASD. And there is quite a lot of work going on currently to try and understand what are the phenotypic differences between high-functioning males and females that lead to females not receiving a diagnosis in childhood. My clinical impression is that these very able females are often more interested in social interactions than men, and quite often their restricted interests are not unusual and sometimes not as intense as in men.
Thank you. In regarding twin studies, recent studies have shown a higher rate of concordance in dizotic twins with respect to ASD. What are your thoughts on this?
Well, there are not a very large number of twin studies. Most of the studies continue to show big differences in the concordance rates between identical and non-identical twins. There is always a problem in twin studies of what is known as ascertainment bias. And this is the tendency for concordant pairs of twins, whether they are identical or non-identical, to preferentially be recruited into studies. So there was a recent study from the US that found concordance in non-identical twins of 40%, but when one read the fine detail of the methodology, it became obvious that there had been or appeared to have been some ascertainment bias because a relatively small proportion of the total twin population had actually entered into the study, and that's the sort of situation in which one expects over-inclusion of concordant pairs. A good way of knowing whether a twin study is representative or not is whether the concordance rate in the DZ pairs is comparable to the recurrence risk in siblings. So one would be expecting recurrence risks of the order of 6 to 10% currently, and that would be the sort of concordance rate one would expect in DZ twins if DZ twinning was not in itself a risk factor for ASD.
Thank you. And in thinking about, as you mentioned in your presentation, looking forward and what we should be looking to in the future, do you think that changes to the ASD diagnostic criteria interplay with the results of genetic studies? How do you see that relationship playing out?
So I don't think that the recent changes in DSM-5 are going to have any material effect on genetic studies. I think one thing we need to be aware of is that we have a sort of Chinese menu approach to the diagnosis of ASD, in that individuals can obtain a diagnosis showing widely differing combinations of clinical characteristics. And that's why I think there needs to be increasing emphasis in the future on some fine detailed phenotyping of those individuals who carry recognized susceptibility variants, in case we're able to see some consistent pattern of an association between a genetic risk factor and a particular clinical sub-pattern within the broader range of ASD.
Thank you. If the majority of ASD is not monogenic, how can we use genetic testing to determine combinatorial impact, or the combination of gene interactions?
Sorry, Rachel, could you repeat that, please?
Of course. So if the majority of ASD is not monogenic and there's essentially multiple genes at play, can you clarify how genetic testing might determine those kind of combinative impacts?
Okay, so one of the things I didn't mention, because it's sort of a minor point, is there have been some groups that have identified individuals with ASD who have mutations in two quite different genes. This is a sort of example of a combination of genes, but the total number of people in the literature who have identified mutations or changes in two or more genes is extremely small. So at the moment, we really don't know how to think of combinations of genes. The way geneticists will go about this in the future is, within a particular sample, they will take out from the analysis those individuals who have an identified mutation. So the overall sample is getting smaller and smaller, and you hope that you're, in a sense, removing what I'm going to call noise, in the hope of identifying genes that predispose to autism in the bulk of cases. An alternative strategy is you take those individuals who have a mutation in a gene that you think is not sufficient cause for autism, but is a contributory risk factor, and then in a group of those individuals, genotype them looking for other genetic risk factors.
Thank you. What's the current opinion regarding clinicians using pharmacogenetics to guide ASD patients on effective pharmacological treatment for the issues that they're seeking treatment for, such as including ASD symptoms as well as comorbid symptoms of anxiety, depression, OCD?
I'm not sure what the current opinion is in the general sense. I have my own opinion, which is I don't think that we currently have sufficient information for that to be a practical approach in any more than a handful of individuals. I think what people have to bear in mind is that most of our information about the genetics of autism is from people involved in research studies. Most people with ASD in the world do not receive clinical array CGH testing for genes. So in the vast majority of people with ASD, we have no molecular genetic information at all. Clearly, the people who do get genetic testing tend to be those who are dysmorphic or have adverse drug reactions or have epilepsy or severe intellectual difficulties. So it is possible that in the future, some of those people may receive very personalized care, but I think for the bulk of individuals, that possibility is still a long way in the future.
Thank you very much, Dr. Bailey, and thank you to our audience for excellent questions. I'd now like to turn it over to Laura Holmes, who will be asking Dr. Bailey for his insight into career and professional development.
Hi, Dr. Bailey. I wanted to start out by just asking if you could tell us a little bit about your career to date. I know that's a really broad question.
So I started life studying pediatrics after medical school, and it was suggested to me by several of my consultants that I should think about a career in child psychiatry, and they recommended that I go and study with Michael Rutter at the Institute of Psychiatry. So after a few years of general psychiatric training there, I joined the final stages of the second UK twin study of autism. At the time, thinking that that was something I would just do for a year and go back to what I thought was my main area of interest at the time, which was psychosis in adolescents. And it was whilst I was doing the twin study that Michael Rutter persuaded me to stay in the field of autism, and that's back in 1988, I suppose. So really, I fell into the field of autism almost by accident, and I was particularly interested in the brain base of autism, so I set up a brain bank for autism at the Institute of Psychiatry, because at the time there were very few post-mortem studies of autism, and the supposition was that this was a disorder highly localized to the amygdala and hippocampus, and I and many others wasn't sure that this was a proper explanation, so it seemed sensible to set up a brain bank, because at the time there was really only one brain bank in the US, so that was how my interest in post-mortem work became established. And in terms of brain imaging, I started by doing some MRI structural imaging, and then became aware of a technique known as magnetoencephalography, which provides real-time electrical data from the brain, but with much greater resolution than EEG, and saw that as a particularly useful mechanism for studying autism, because it didn't involve application of electrodes, very user-friendly, quiet machine in which you sit upright, but we didn't have a MEG scanner in the UK at the time, so I started taking adults with ASD over to Helsinki in Finland, where they did have a scanner, and that led on to studies of children with ASD in Finland, and eventually to having our own MEG scanner in Oxford. And the work on phenotyping of autism that was really pioneered by, or phenotyping other relatives pioneered by Rutter and Folstein, naturally led on involvement in molecular genetic studies that were in strong collaboration with Tony Monaco, who was already a very well-established molecular geneticist at Oxford. Since I moved to Vancouver, a lot of my work has been in more applied aspects of research, particularly focusing on adults, but I'm hoping that we will get back into genetic research, particularly focusing on individuals with ASD and comorbid epilepsy.
Wonderful. What is one thing in your own training that you think helped you to be successful that our participants here should seek out?
I think the biggest factor contributing to modest success in my career was being at the Institute of Psychiatry, which is one of the world's great centers for psychiatric research, and particularly having a very good mentor in the shape of Michael Rutter, who brought on many of the current leading autism researchers in the UK. I think having a wise and experienced mentor is absolutely key, not just in the early stages of one's career, but even later in one's career as well.
Is there something that makes a trainee stand out to you as someone that you would want to work with or mentor?
I think I'm always interested in very smart people who have ideas, so those sorts of people are always attractive. But I think also people who are mature and have a well-balanced personality means that one's likely to be able to have a productive long-term relationship with the person as well.
Is there a hard-won professional insight that you tend to pass on to people that you mentor or advise?
Persistent.
An insight.
Yes, no, I think sometimes success in science is just about hanging on in there and being persistent. Science is
I see.
An occupation that involves extreme delay of gratification and goals, and so sometimes the payoff from scientific research can be many years later, and I think the funding climate for biological sciences, particularly clinical aspects of biological sciences, is not great at the moment, and so people have to be able to weather, I think, periods of time when funding is limited and know that at some time the winds will change and things will get better. So stickability and persistence, I think, are absolutely crucial traits for a long-term career in science.
That is good to hear. Is there an existing gap in your field that students and trainees might not think to consider?
Well, as I suppose was sort of implicit in my presentation, although there have been exciting developments in the genetics of autism, we are not at a stage where we had hoped to be in terms of having insights into the neurobiology that would lead to effective biological interventions. And so I think there is a real need to be developing more effective non-biological interventions, and particularly to realize that the majority of the people in the world with ASD are adults, not children, and that we have very few interventions tailored towards adults. And indeed, it's a sad state of affairs that worldwide most adults with ASD receive little or no clinical care at all. So this is really the sort of gaping opportunity both for research and clinical provision.
What is your most successful technique for networking with other professionals? I know this is an area that students sort of struggle to get the hang of sometimes.
I'm not a terribly good networker because I tend to suffer from social anxiety. So if I go to a new place, I will hunt out someone I already know, and not terribly good at talking to people I don't. In my experience, the networking comes about if you give a good talk. People will come up to you afterwards, want to talk to you, might then say, oh, let's have a drink or let's have dinner. So I think good presentations or good posters or the ways to make those important links, at least for me.
Oh, that's great to hear. Do you have one best piece of advice that has stuck out to you that you've received?
I've had a couple of good ones. One I think has been always to be open to collaboration. That science is often a rather solitary pursuit, but increasingly, as we're trying to tackle big and complex problems, we often can't do it either in our own lab or in our own field. We need to collaborate with people with complementary experience. So I think being open to collaboration. And then a very wise professor of child psychiatry in Europe once said to me that he'd realized that if he ignored problems, a lot of them eventually went away. And that one needs to be very clear in everyday life when faced with scientific or clinical problems, which are the ones one's really got to try and solve and which ones are merely a time-consuming distraction that probably will disappear into the long grass eventually.
I like that. We had one courageous undergraduate who asked if you had any particular advice for undergraduates interested in pursuing research in biological mechanisms of autism.
Do I have good advice? I'm not sure it's good advice, but I think one advice would be to spend some time with people with ASD, because often some of the best insights and ideas that people have had have simply come from observing people with ASD. So there's a limit to what we can come up with just on bits of paper or reading scholarly articles. We need to spend time with people with ASD to have a feel for the disorder and to generate our own ideas. And then I think my second piece of advice would be, if that is an area that people are thinking of pursuing in the long term, then to make sure you head for a lab that already does ASD research or is next door to a lab that does. You want to bring yourself into proximity with people who are currently doing ASD research.
Thank you.
Great. Well, I think that we are running out of time, so we'll go ahead and wrap this session up. Sorry, this is Carolyn again. And I'd just like to end this session by thanking Dr. Bailey, the working group, and everyone for joining us live today. Just want to remind you that the video replays are available to INSAR members for the entire 6-series course, including this one. You can become an INSAR member by visiting autism-insar.org. If you have any questions or want to get more involved, you can contact the Student and Trainee Committee at studentcommittee@insar-autism.org. We hope that you've enjoyed this first session of the 2016 INSAR Summer Institute series. Please join us next week when Joe Piven will discuss infant siblings of children with ASD. Thank you so much.